Hello, welcome to another boring and probably not very useful video about prediction of antidepressant response to this probably antidepressant by norepinephrine transporter transgene polymorphism. So the names look quite Japanese and they must be from Japan. With a multitude of antidepressants available, predictors of response to different classes of antidepressants are of considerable interest. So they try to create an individual's treatment based on the genetics, and they're using gene polymorphism for doing such research. However, those studies are not really that solid. Like, you think as a genetic study it has to give the same result, but there are many different studies finding different results because of many reasons. So here we see the purpose is determine whether this gene and serotonin transporter gene polymorphisms are associated with antidepressant response to this Milnacipran, not sure how to pronounce, a dual serotonin norepinephrine reuptake inhibitor. So this is the method. 96 Japanese patients with major depressive disorder treated with this antidepressant with this dose for 6 weeks. It's a short term, actually not very long term. Severity of depression was assessed with this scale, and the method of polymerase chain reaction was used to determine allelic variance. 80 patients completed the study. The presence of the T allele of this polymorphism was associated with a superior antidepressant response. That's interesting. It's important to see the effect size. The results suggest that NET but not 5-HTT polymorphisms in part determine the antidepressant response to Milnacipran, and it's important to find a significant result using categoric variables, because SPS is inclined to give a lot of fake positive results, or like there's no actually that much association. But here, first, the 96, only there are 96 people, and second, they're using probably categoric variables to code the genetics, to code the polymorphism. So that's why it's very interesting and very important study, and there is a risk that they cannot find any significant result. At that time, no journal will be interested in publishing the results, which is a problem. So they mention about regarding genotyping, how they genotype, and statistical analysis. We can read this. T-test or chi-square test were appropriate. So as I said, categoric test. So it's hard to find significant result, because I think 1 and 0 coded there, or 1, 2, 3, 4, maybe it can be coded like that, the polymorphisms. So as I said, genotype distribution and allele frequencies were analyzed with the use of chi-square test and power analysis. So they also did power analysis. It seems to be because the sample size is not very large. So if you're more interested in reading this study, you can find this. You can search this title and you can read this article without needing any much effort to, sometimes journals like to hide full article. And thanks for watching this video. I hope you enjoyed this article, or the way that I present this article. Have a beautiful day. Bye bye.